All right. So when we last met, we started talking about the cell cycle and cell division and why this is an important process in organisms. We know that our bodies are constantly undergoing cell division, and we looked at one of the first types of cell division, which was mitosis. And we now understand that mitosis is required for our bodies to continue to just reproduce these normal somatic cells, right? These somatic cells are responsible for basic processes in our bodies like growth and development and just maintaining the organism, right? So we said that the cell cycle contained two main phases, which were interphase and M phase, right? Collectively speaking, we said that interphase was characterized by periods of preparation, growth, metabolism, enzyme production, duplicating the DNA, and so forth, and preparing the cell to divide. The main thing that we take away from the cell cycle and this process of mitosis is that we end up with two genetically identical daughter cells being made, right? And so it makes sense that one of the phases in the cell cycle is that we have to duplicate the genetic material that's already there, so that when we create these two new, when that cell splits in half, both new daughter cells have the exact same genetic content. So we're essentially making a duplicate. All right. Sometimes we refer to this process as cloning. So anyway, we're going to push through here. We've already talked about what's happening at each phase of the cell cycle, G1, S, G2, and M phase. And we said that M phase had two main things going on, right? Mitosis, which is basically some more preparatory steps for division. And then cytokinesis is the actual division of the cytoplasm of that parent cell into two new cells. Yes, so when we're getting ready to talk about meiosis in just a second, Ms. Bennett, but that's exactly what's happening. It's basically, it's like mitosis happening twice, right? But it's what we refer to as a reductive cell division, because instead of getting a daughter cell that's going to have 46 chromosomes, it's actually going to have 23. So that's exactly where we're going today. And so anyway, this was just another illustration that shows you what's happening during the cell cycle. G1 is that sort of preparatory phase where the cell is growing and everything, but we understand there's no cellular division taking place in G1, right? S was our synthesis phase, where we're basically synthesizing new DNA or duplicating the number of chromosomes that are present in the cell. And then G2 was another gap phase that's making sure that all things have happened sort of with fidelity and so forth before the cell progresses into M phase, okay? And so then we highlighted the stages of mitosis, which were prophase, metaphase, anaphase, telophase, and cytokinesis, where that cytoplasm actually divides and produces two daughter cells. All right, so I showed you a few illustrations. I won't spend much more time on it. You had a couple questions for homework. And again, here are just the text explaining what's happening in mitosis. We said that the first stages of mitosis was prophase, right? Where these chromosomes begin to condense and compact and become visible. Those sister chromatids are going to be visible now. We know that there's a little component of these of this chromosome known as the centromere that sort of holds these two sister chromatids together. So you've probably seen this illustration before, right? Where we kind of have this X-like structure with this circular structure in the middle. That's our centromere. And so the next phase would be metaphase, where we would see the chromosomes sort of align towards the midplate or the middle of the cell, okay? At this point, again, we're checking to make sure things are there, that the number of chromosomes that should be there are there in their normal pairs, okay? Anaphase is the third step of mitosis. We talked about this already, where these sister chromatids begin to separate and sort of start moving towards opposite poles. Because remember here, the end goal is that we have two distinct groups of chromosomes at either end of the cell, so that when the cytoplasm splits in half, we have two new cells that are going to have the same exact genetic material, okay? So anaphase ends when all chromosomes have reached the opposite poles. During telophase, the chromosomes have arrived at opposite ends of the pole, and you start to see sort of this nuclear envelope forming around each new set of chromosomes. And again, this is leading us towards the next step, which is that cleavage, right? That final step of M phase is going to be called cytokinesis. And what we start to see is sort of this indentation. I'll go back a slide. You start to see the cytoplasm starting to indent a little. Okay, that's an indication that this cell is ready for division, is ready for cytoplasm division. Okay, cytokinesis. And that cleavage furrow forms, right? The cleavage furrow is characterized by the indentation of the cell surface in order to create a pathway for separation of those two daughter cells during cytokinesis. And so here's what you should have in your notes. As a big picture here, the end result of mitosis, it's going to be two cells that are genetically identical to the parent cell. So whatever the genetic makeup is of the parent cell, that's going to be the genetic makeup of that daughter cell, okay? Each daughter cell, put this in your notes, each daughter cell receives exactly the same number and kinds of chromosomes that the parent cell has, okay? So the result of mitotic cell division are going to be two genetically identical daughter cells that are going to have the same type, same number of chromosomes that the parent cell had. Okay. And so that's what happens during mitosis. The other type of cell division that was, and so purpose, right? Purpose of mitosis is to produce new somatic cells or body cells that are going to be involved in just that general, maintenance, growth, so forth, repairing damage, just sustaining the organism over time. That is the purpose of mitosis. Now, meiosis has a completely different purpose, okay? The other type of cellular division that we're going to talk about today is meiosis, okay? The main reason that we need meiosis is for sexual reproduction. Write that down. So there are a couple things you should grab by the end of this talk regarding sexual reproduction. Okay, there are different types of sexual reproduction. We want you to be able to differentiate between asexual and sexual reproduction. And this is probably going to be a review for you. You've heard this before in your high school biology class. All right, so we want to be able to distinguish between asexual and sexual reproduction. We want to be able to distinguish between haploid and diploid cells, okay, and this will be determined by the number of chromosomes that are present in these cells. We want to define the term homologous chromosomes. All right, we want to explain the significance of meiosis and describe what's happening. And as Amari sort of mentioned earlier in our chat, meiosis is basically mitosis happening twice, right? It's going to be 2 rounds of cell division. And the significance here is that the cells that are being produced are going to be gametes, okay? Not somatic cells, but sex cells are produced during meiosis. That's a very significant difference between mitosis and meiosis, okay? Mitosis produces body cells. Meiosis produces sex cells or gametes, okay? And so you should certainly be able to sort of compare and contrast mitosis and meiosis, emphasizing the different outcomes, and we'll take a look at that today. And so we're looking at reproduction here, right? So 2 basic types of reproduction that are in eukaryotes, right? Asexual reproduction and sexual reproduction, okay? Asexual reproduction requires only a single parent, all right? And so what happens is that single parent can undergo sort of a budding or splitting or fragmentation process to produce, 2 or more new individuals being created. All right. And this form of asexual reproduction is mostly due to this mitotic cell division. New individuals are going to inherit the genes and traits that are going to be identical to that of the parent, right? So there's only one parent involved in asexual reproduction. Okay, there are some advantages to this process, one being that it can occur very quickly. It's a pretty rapid process. It typically happens efficiently. The organisms don't require a mate. So again, there aren't many outside hindrances from this process taking place, right? However, there can be some disadvantages to this type of process, particularly when you think about the genetic outcome. So we're saying that the new organisms that are generated through asexual reproduction, through budding, binary fission, whatever type of reproduction it may be, the offsprings that are being made are going to be genetically identical to the parent. So what could be wrong with that picture? Well, if there are any type of genetic abnormalities in the parent, guess what? That's going to be passed down to the offspring. Okay, so while it happens rapidly and pretty efficiently, you're going to transfer any abnormalities, genetic abnormalities as well. Okay. In contrast, sexual reproduction requires two parents. All right. Sexual reproduction is characterized by the union of two sex cells, right, that are called gametes. So we're talking about the sperm and the egg here, right? Two different parents contribute to the formation of these gametes, right? So for the human, we're talking about mom and dad, both contributing genetic material for the formation of this new individual. These gametes are going to unite to form a single fertilized egg that we'll call a zygote, right? So we're getting some new terms here in blue. Make sure you're jotting down these terms. Right? We use the term gamete to refer to our sex cells, right? The sperm and the egg. A zygote is the result of unification of the sperm and egg cell becoming fertilized. A zygote is formed as a result of fertilization. All right. So asexual sexual reproduction, two processes in which new individuals are formed, but the outcomes are going to be different. From asexual reproduction, you're going to get new individuals that are going to be genetically the same as the parent. So we sometimes refer to this again as cloning, right? Whereas sexual reproduction is going to produce genetically different offsprings, right? Because you're mixing genetic material from two parents in this case. Okay, so that offspring that's made is going to be a hybrid between both mom and dad, right? So the results here, make sure you jot this down in your notes. Asexual reproduction produces genetically identical organisms that we'll call a clone, right? Whereas sexual reproduction results in genetic variation where the offsprings are not genetically identical to the parent. Because in this case, we've got the joining or the union of genetic information from two parties, right? So the offspring becomes a hybrid, if you will. Okay. What questions do we have so far? Perfect. Visual. I know some of us are visual. If we take a look at this panel over here on the right, asexual reproduction, we start with one parent. It's going to have a genetic content. The offspring for asexual reproduction is going to be identical to that parent. It's going to be identical, genetically identical to the parent. In contrast, sexual reproduction, we've got the joining or union of genetic information from two parents, okay? We've got mom, we've got dad represented here in blue, mom represented here in pink. This offspring is basically gonna manifest as a mixture of both parents genetically, okay? You should definitely be able to describe and distinguish between these two modes of replication, of reproduction, okay? How many parents are involved? What does the offspring look like? All right. So some general characteristics here, some points of comparison. Sexual reproduction requires two organisms. Okay. In the case of the human, we're going to have the sperm and egg fertilizing to form a zygote. The offsprings are going to be genetically unique or different from the parents, okay? But both are going to contain a random combination of parental genes, right? So there's no rhyme or rhythm as to which genes for which characteristics you're going to get from mom or dad. This is a random assortment of genes here that's happening during crossing over, during meiosis, and we'll take a look at that in just a second, but it's all random. This form of reproduction is used by humans and other animals, right? Advantages, pros to sexual reproduction is that you're going to be able to produce genetically different babies. Okay, and what this is doing is providing a survival advantage, right? So you're in theory, by being genetically different, right, you're inheriting the ability to survive, increasing your chances of survival, right? Some of the disadvantages of sexual reproduction is that it's time consuming. All right. It requires a lot of time, a lot of effort. You've got to have both parties present. It's got to be just, optimal conditions for it to take place. Like, it's not a very easy task to accomplish. It takes a very long time in order to, create this new individual and undergo development and so forth. Right. On the other hand, asexual reproduction only requires one parent, right? This single organism is basically going to make a copy of itself and sort of divide into two, all right? The offsprings are genetically identical to the parent. Organisms that use this form of reproduction include yeast, sponges, bacteria, right? Pros of asexual reproduction, as I mentioned, is a pretty efficient process, right? It happens quickly, typically efficiently, without too many problems. However, a disadvantage again is that if the parent has any genetic defects or deficiencies, all of those would be transferred to the offspring. Okay, so make a note of this. Okay. Any questions about distinguishing between sexual and asexual reproduction? So when we're talking about the genetic makeup, we have what we call homologous chromosomes, right? Each chromosome in those somatic cells, right? The somatic cells are going to be our body cells. All cells other than our sex cells are going to be somatic cells, right? So each chromosome in a somatic cell normally has a partner, right? Two partners are known as homologous chromosomes. They're going to typically be similar in size, the position of that centromere. They're going to carry the same genetic traits or genetic information. This is often what you see when you're looking at your karyotypes, right? Those homologous chromosomes. And we're saying that homologous chromosomes are going to code for the same genes, right? These same genes, we know that genes are, we talked about the gene as a segment of DNA that codes for particular traits in an individual, all right? And so that's where we'll find these same genes in the same location on our homologous chromosomes, all right? So we're talking about chromosome number in our somatic cells. If a cell has two sets of chromosomes, it is said to have a diploid chromosome number. Write that term down, diploid. Diploid means that you have two copies of these chromosomes, right? Diploid chromosome numbers are what we find in our somatic cells, right? Our somatic cells are going to have exactly two sets of each chromosomes in pairs. Chromosomes number 1 through 22 on the human karyotype are going to be diploid, all right? In humans, the diploid chromosome number is 46, all right? All of our cells, all of our somatic cells in a normal human are going to have 46 chromosomes, right? However, if the cell only has one set of chromosomes, it is said to have a haploid number. So haploid meaning half. So what types of cells do we expect to have a haploid number of chromosomes? That's a question.
Can you repeat?
What types of cells in our bodies would we expect to have a haploid number of chromosomes?
So the sex cells?
Sex cells, absolutely. Why? Why does that make sense for you, Amari?
Because if your sexual reproduction then you would your sex cells plus somebody else's would combine.
Absolutely. That's absolutely what's happening. Okay. During sexual reproduction, these gametes or these sex cells, right, are going to have a haploid number of chromosomes because it's going to join with another sex cell, right? Conceivably the sperm and the egg. These are our sex cells in humans. They're each going to have 23 chromosomes. That is a haploid number of chromosomes, right? So write that down. The types of cells in the human that would have a haploid number are gametes or sex cells, right? Whereas our somatic cells are going to contain a diploid number of chromosomes. Okay. Right? So this is just a visual for those of us that are visual. Cells that are going to be diploid are going to have two copies, two sets of each chromosome. Haploid chromosomes are only going to have one or half the number of diploid. Okay? So what happens during fertilization? We've already said it. When a sperm and an egg fuse during fertilization, each gamete is haploid. Okay, each one of those sperm and egg are going to contribute one set of chromosomes, right? And we therefore see a restoration of that diploid number in a zygote. Okay, and so during mitosis, this zygote is going to divide to form the first two cells of the embryo. Each daughter is going to receive the diploid number, which is 46. Somatic cells are diploid. You should have that in your notes. Somatic cells have a diploid number of chromosomes. Another visual here, okay? Diploid cells are going to have two copies of each chromosome that's present, okay? Those two pairs of homologous chromosomes are going to have the same information on them, the same genes, all right? Haploid number, there's one copy of each chromosome, okay? So we sometimes refer to the diploid as 2N, and N being the number of chromosomes. Haploid would just be N, all right? So meiosis. We said the sole purpose of meiosis was to produce these sex cells, right? Meiosis literally means to make smaller, right? So meiosis is a reductive cell division, okay? This is a type of cell division that reduces the number of chromosomes by 1/2, right? A diploid cell basically undergoes 2 rounds of cell division, yielding 4 haploid cells, right? So if you had to visualize what's happening during meiosis, you're going to think about what you just learned for mitosis, but that's going to happen twice. Okay, so we're going to have 2 rounds of cell division that are going to produce 4 daughter cells instead of 2, right? So let's see. During meiosis, there's a process known as crossing over that takes place. And what's happening during crossing over is that these chromosomes are going to sort of separate and disperse to these 4 haploid daughter cells in a very random motion. All right, you'll get some more information about this when we get into our genetics unit and when you actually take your genetics course. But you should understand from a basic standpoint right now that sexual reproduction and this process of crossing over is how we end up with offsprings or siblings that look different, right? Because each time mom and dad procreate, each time this sperm and egg unites, there's going to be a random disbursement of genes from both mom and dad into these daughter cells, right? And so meiosis, there's 2 rounds of cell division, right? During meiosis 1, 2 haploid cells are made from a diploid cell. And then during that second round of cell division, those chromosomes are going to separate into sister chromatids, and the number of chromosomes present in each cell is going to reduce. And we're going to end up with 4 haploid daughter cells, right? So in summary, what I want you to understand about the events of meiosis is that there are some similarities and differences for mitosis and meiosis. Okay. Meiosis involves 2 rounds of cell division, right, that produce up to 4 daughter cells. Write that down. However, though there are 2 rounds of division, the DNA is only going to be duplicated once. Okay. Each of the 4 cells that are produced by meiosis are going to have a haploid number of chromosomes, meaning they're going to have 23 chromosomes, right? Because we're producing gametes. Okay, gametes can only have 23 chromosomes. And each haploid cell is going to have a unique combination of genes from mom and dad. So all of these daughter cells that are being produced from meiosis are going to be genetically different. Okay. And I see a typo here on this slide. That first bullet should be TO and not TWO. So we're saying that the events of meiosis are very similar to what's happening in mitosis, but there are 4 important differences. The first one being there's going to be 2 rounds of cell division, right? But we're only going to duplicate that DNA once, right? Each of the 4 cells that are produced are going to contain a haploid number instead of a diploid number. And each of those haploid cells are going to have unique combinations of genes instead of being all the same. All right. So do take some time, this evening to come back and synthesize this information and compare these 2 types of cell division, meiosis and mitosis, their purpose, what's happening, and what the outcomes are for each process. This is a visual here of what's happening during mitosis and meiosis, okay? For mitosis, we know that we're starting with a diploid cell, right? This process of mitosis will occur, right? The stages of mitosis are prophase, metaphase, anaphase, telophase, and cytokinesis, and we end up with 2 diploid daughter cells that are going to be genetically identical, okay? However, meiosis is a little bit different, right? We're going to start with a diploid cell, right? It's going to undergo 2 rounds of cell division. So we've got one where it sort of just looks like what's happening during mitosis, where we duplicate our DNA, prepare for division, and undergo the steps of prophase, metaphase, anaphase, telophase, so forth. However, we don't stop there, right? There's another round of cell division, right? That's ultimately going to provide us with 4 genetically identical haploid cells. Okay. So let's stop here. For homework, oops. For homework, in your notes, you're going to describe the significance of meiosis. Why is this an important process for organisms? Okay. Describe what's happening during meiosis. In your notes, think about, read back and compare and sort of contrast what's happening during mitosis. How is meiosis any different? Okay. So you should understand the difference in terms of the purpose or the significance of this cellular process and what's happening. All right, let's see. So we said the meiosis is required to produce our egg and sperms for sexual reproduction. That is the purpose or the significance of this cellular process, okay? So the sole purpose of meiosis is to produce gametes. Okay, and so the goal here biologically is to produce daughter cells with half as many chromosomes as the starting cell, right? So we saw in our visual a few slides back that we start with that diploid parent cell here, and then we're going to undergo 2 rounds of cell division where we end up with daughter cells that are going to have half the number of chromosomes than that of which it started. All right, this is just kind of me repeating what I've already said. The phases of meiosis, there are two. This is a two-step division process. It's a reductive cell division. We start with 46 chromosomes. We're going to end up with cells that only have 23. So those two steps are meiosis I, meiosis II. All right, we start with that diploid cell. All right, we're going to go that first round of cell division. We're going to have two diploid cells being produced. All right, and then we undergo that second round of cell division where we're now reducing the number of chromosomes to 23. All right, so we end up with 4 haploid cells being produced from meiosis. Okay, as I said before, cell cycle starts as it normally would. Okay, the process of meiosis begins with prophase 1, metaphase 1, anaphase 1, telophase 1. These are all the same steps that appeared in mitosis, right? Meiosis II, you have another round of cell division that are ultimately going to result in 4 haploid cells being produced. Meiosis is important, okay, because it generates genetic diversity. Genetic diversity is what we need for increased chances of survival, okay? And so what happens is that the exchange of genetic material between these homologous chromosomes in meiosis I leads to, again, these random assortments or alignments of disbursements of chromosomes from both mom and dad, right? That leads to genetic diversity. Some concept review questions that I want you to work on at the end of today are explaining the significance of mitosis, explain the significance of meiosis, okay? Identify 3 to 4 distinct differences between these two cellular processes and describe what happens during crossing over, all right? The concept review questions